but my job for the next 20 minutes is to give you an overview of the top 10 basic science papers of 2018. I have no relevant financial disclosures to this talk. I would put in a couple academic disclosures, which is that the top papers, or ranking of the top papers, is somewhat subjective, and I tried to base it at least somewhat in objective data using citation frequency according to Web of Science and Scopus and these other tools. And then I combine that with my own impression of hot topics within the scientific community. So it's a little biased toward lung cancer, which you'll pick up as we move through for both of those reasons, but I tried to include some other papers as well. So perhaps one of the most talked about papers from this year was a paper from Cell describing a drug that specifically inhibits the mutant version of KRAS, specifically the G12C mutation. As you know, KRAS is the most frequently mutated oncogene in lung cancer, and the G12C mutation in particular is the most commonly identified allele. And previously, blocking KRAS was very difficult. It was called undruggable because KRAS isn't a kinase in the traditional sense. It doesn't have an ATP binding pocket. It's not amenable to these other strategies that we've used over the years to inhibit these proteins. It exists in an active GTP-bound state and in an inactive GDP-bound state. So chemists in this paper created a compound that covalently binds to the inactive form of the mutant allele specifically and locks it in that inactive configuration. The drug is orally bioavailable, so you can take it orally, and it has minimal toxicity because it only really impacts the mutant protein. So they show both in vitro and in vivo data regarding efficacy. This is the H358 cell line over here is a lung cancer cell line with the G12C mutation. You can see in the gray and the red curve there that the cells are sensitive. When they re-express a mutation that's not G12C, like G12V, the cells become resistant. So it really is specific for that particular mutation. They did show downstream that there are mechanistic consequences to blocking RAS in that setting. You see decrease in phospho-ERK and increase in apoptotic proteins like cleaved PARP, suggesting that the drug is working on target. You see similar data in vivo. So these are the top is flank xenografts, both in a pancreatic model, so that MIAPACA2 is a pancreatic cancer cell line with a G12C mutation. The 441 is a lung cell line with G12V. So in the setting of G12C, you can see at 2 doses of the drug ARA-1620 that those tumors are inhibited, particularly in the higher dose, which is that red line, whereas in the G12C cells, they basically grow as if they're unaffected. And they show the same thing in PDX models, so patient-derived xenografts with G12C mutations are predominantly sensitive, and then when they look at the G12D or wild-type KRAS, those tumors really don't respond. So this is sort of an exciting area for treatment. Topic number 2, osimertinib. So osimertinib is a third-generation EGFR TKI, and it's effective in cases of the T790M resistance mutation. It's become really the first-line therapy for the majority of patients with EGFR mutant lung cancer, and despite its efficacy over time, over the course of a year, so patients develop resistance, and there's a lot of efforts now to understand why that is. So this is a group from Dana-Farber who did whole genome sequencing both on pretreatment samples and then samples that had progressed to try and identify relevant resistance mutations. So they found that they could break those down into 2 major groups. The first was those tumors that maintain the T790M mutation, and that was about 1/3 of the patients. And of those patients, the majority actually had a second mutation at a site called C797S, which generated resistance to osimertinib. The second group was those with T790M loss, which was actually the majority of patients. And there were a number of resistance mechanisms they saw in that group: small cell transformation, amplification of MET, mutation of other oncogenes that generated resistance. They saw the patients that lost T790M, so that larger group actually had a shorter time to disease progression, so they tended to fare worse. And the T790M status and the status of some of those other mutations I showed you was actually detectable in plasma in those patients, suggesting that circulating tumor DNA might be a means to identify resistance mechanisms. And maybe what I thought was most interesting was they went back to those pretreatment samples in patients with those resistance mechanisms, and for the most part, they can identify small populations of cells in the pretreatment setting that had those resistance mechanisms, and those cells actually grew out over time. For instance, In the cells that had small cell transformation, they went back to the original population. They could find a small number of cells that actually had RB loss already, even at the time of presentation. And over time, those cells grew out because of selection. Topic number 3, so SHP2. There were 4 papers this year in major journals looking at SHP2 as a therapeutic target in cancer, particularly in lung cancer. So SHP2 is a tyrosine phosphatase, so it cleaves phosphates from various proteins, and it sort of functions between the receptor at the cell surface and the KRAS machinery downstream to facilitate KRAS signaling. The mechanism by which it does that is relatively unclear, but what these folks found is that if you inactivate SHP2 in the setting of RAS activation, that the tumors progress more slowly. So that's the curve on the left there. So in the traditional KRAS P53 mouse model, those cells are shown in blue. And then if you lost SHP2 genetically, those cells are green. Those mouse survive significantly longer. Similarly, on the right side, they did it pharmacologically. So they blocked SHP2 with a drug, the GS493, gave it in combination with trametinib, which is a MEK inhibitor, shown there in green, and they showed significant tumor regressions in the combination-treated animals. And they apply that to a number of different scenarios. So ALK resistance, so if they took cell lines that were sensitive to ALK inhibitors that eventually became resistant, and they combined then ALK with SHP2 inhibition, they resensitized the cells. And it's interesting because SHP2 doesn't necessarily facilitate only the signaling through one RTK, it's through a broad range of RTKs, so it sort of covers whatever the potential resistance spectrum might be. The curve on the right there is sort of the sole paper from esophageal cancer that I included in this talk, which also showed that in the setting of KRAS amplified esophageal cancer, if you combine MEK inhibition, so inhibit KRAS through MEK, and combine it with SHP2, that they saw tumor regressions in animal models of esophageal cancer as well. This is a paper I like a lot regarding STK11. So there's some evidence now to say that STK11 mutation impacts one's sensitivity or resistance to immunotherapy. So STK11 is a tumor suppressor that's mutated in about 20% of lung cancer. It's really interesting in the sense that it functions as a serine threonine kinase that regulates cellular metabolism. But when you inactivate it, there are metabolites that build up that then facilitate DNA methylation. So it's involved both in cellular metabolism and epigenetic gene expression. So these guys looked at several clinical cohorts to suggest that there were low response rates in KRAS mutant STK11 mutant tumors relative to KRAS P53 tumors, and that's shown in the survival curve on the top there. So blue being KRAS P53, so wild type STK11, and the red showing decreased survival in the KRAS LKB1 or STK11 mutants. They also found that it was particularly relevant in cases where patients had low PD1 but high tumor mutation burden, suggests maybe that it suppresses immune responses in that particular state. Then they validated it experimentally. So they took mouse models either with KRAS activation on the left side, so you can see with just activated RAS and functional STK11, when you treated those mice with anti-PD1 treatment, which is the red line, their tumors were suppressed. When you took those same mice and you inactivated STK11 in the setting of activated RAS and you treated them with PD1 drugs, the tumors didn't respond at all. So that was pretty striking data. There was a follow-up paper actually from this past year, 2019, showing that loss of STK11 decreases STING expression. STING is a molecule that's really important for interferon signaling. And they think that it's because the methylation, STK11 loss facilitates methylation of these various loci, one of which may be STING, to suppress inflammatory mediators like interferons. This is a really interesting paper. So there's some data now sort of talking about a link between the gut microbiome and your response to immunotherapy. So this group took feces transplants from patients with non-small cell lung cancer who'd been treated with immunotherapy. And they divided them into groups of responders and those that didn't respond. And they took that feces and they transplanted into gut-free mice, so mice that don't have a microbiome of their own. And basically what they found is that in mice that received stool from responders, their tumors actually responded. So you can see here in bioluminescence imaging, the images of the mice on the right. So on the bottom with this flank xenograph, when they got stool from responders and they were treated with anti-PD1 treatment, the tumor basically goes away. In contrast, on the top, when you transplanted the same mouse with the same experimental model with stool from a non-responder, that the tumor is basically totally unaffected by PD1 treatment. So that seemed a little dubious, but they did actually go back into the stool of those patients and do deep sequencing to look at what the representation of bacteria is in that stool and find bacteria that are overrepresented in those responding patients. And they found a couple, one of which is this Akkermansia mucinophila, and the other one is an Enterococcus subspecies, and found that those were much more common in the stool of responders. So you can see then they took, again, a mouse model when they gave it saline, which is that survival curve all the way on the left, the tumors, and they treated with anti-PD-1 treatment, the tumors grew. When they took those same mice and they gave them the combination of those two bacteria, they treated them with anti-PD-1 treatment and the tumors were suppressed. And on the right, in those mice that received bacteria, they had higher levels of tumor-infiltrating lymphocytes, and they also had higher levels of CD4 T cells within the tumor, suggested that there's real immunologic biology here. Topic number 6, circulating tumor DNA. So circulating tumor DNA is seemingly everywhere at this point. People are using it for a number of different things: monitoring treatment response, predicting response to immunotherapy, detecting clinically occult residual disease following induction treatment. This is one particular study where they're trying to calculate tumor mutation burden, which is a predictor of sensitivity to immunotherapy, from circulating tumor DNA rather than from the tumor itself, and predicting sensitivity to atezolizumab in that context. So they performed deep sequencing both of the tumor and of the blood, and they show pretty good correlation between the mutational profiles in those two settings. There was discordance in both directions. In other words, there were mutations present in the tumor that weren't in the blood, and there were mutations in the blood that weren't present in the tumor. And they suspect that that's because the blood potentially collects a heterogeneity, it reflects tumor heterogeneity. So if you just sample one area of the tumor, you don't get the whole mutational spectrum, whereas maybe in the blood you get a little bit better of a representation. They went on to show that, that the blood tumor mutation burden did actually predict response to atezolizumab. So in the survival curves on the right, if you had more than 16 mutations per megabase, your progression-free survival curves separated much better than if you had tumor mutation burden less than 16, suggesting that it did work. That's in terms of progression-free survival. The overall survival data is less robust, but it was also significant. So during the last session, there was some talk about deep learning and artificial intelligence, and there are folks trying to apply that to a number of different areas. This is one particular study from NYU where they're trying to apply those algorithms to the interpretation of pathology slides and tumor histology. So I'll admit that my understanding of this is minimal. However, what I can say is that they're using a platform that was developed by Google in 2014 for a competition where they were trying to develop new visual recognition software, and it uses technology that's a little bit different, I think, than what people were doing before. But in this particular case, they trained this Google Inception v3 algorithm with over 1,600 slides from the TCGA, and then validated, once the algorithm was trained, they validated it in in-house specimens from their own institution. And essentially what they find is that the model was accurate for identifying both lung adenocarcinoma and lung squamous cell carcinoma relative to normal lung with pretty impressive sensitivity. It was also adaptable to paraffin tissue, frozen sections, and core biopsies. And in the end, it actually performed on par with human pathologists. So maybe this suggests that pathologists may be obsolete at some point. I don't know. And then I think what was even more impressive was when they went into the data, the software can actually predict the mutational status of several mutations based on the architecture of the tissue. So STK11, EGFR, FAT1, SETBP1, KRAS, and P53 were all predictable with reasonable sensitivity based on that visual recognition. And when they overlaid the mutational pattern with the histology, you could see that those mutations were expressed sort of heterogeneously within the tumor and the tumor microenvironment. So I think this is the sole transplant paper in the talk, but obviously ischemia reperfusion injury is really important in the setting of lung transplant and graft survival. And this is a paper from Dan Krausel's group at WashU where they were looking at new mechanisms of neutrophil infiltration in ischemia reperfusion injury. So neutrophils are one of the effector subtypes of ischemia reperfusion injury following lung transplantation. However, how the neutrophils hone to the tissue and infiltrate the tissue remains relatively unknown. They wrote a previous paper suggesting that non-classical monocytes, so a subset of the monocyte population in the donor lungs, sensed the ischemic injury and then secreted a cytokine called MIP2, which initialize the infiltration of neutrophils. In this study, they sort of expand that model to show that there's a classical, so a larger monocyte population that's resident in the recipient's spleen, and in the setting of ischemia reperfusion, they mobilize from the spleen, go to the lungs, and then secrete things like IL-1 beta, which increases the vascular permeability and facilitates neutrophil infiltration. And impressively, what they did was they actually did, in a mouse model of lung transplant, they did splenectomies to sort of abolish that response. And you can see in the graph on the left, in the setting of splenectomy following transplant, the grafts performed much better, so their PaO2s were much higher, and they had much less neutrophil infiltration. So now they're looking to try and target IL-1 beta or CCR2, which is the monocyte receptor, to generate the clinical therapeutics. So there's some pretty striking evidence now to say that in the setting of KRAS mutation, so when KRAS is mutated, you mutate one allele and you generally keep the wild type copy. And there's evidence to say that retention of that wild type allele acts as a tumor suppressor, actually. And when you mutate that or delete it, the tumors grow much more rapidly. So this is another paper from the Dana-Farber group where they did just that. So this is this graph on the right is a survival curve of mice, either with just KRAS mutation, which is the red line, or KRAS mutation with deletion of the wild type KRAS allele. And you can see in the black line that those mice die significantly faster. So I should say that the mechanism of this is relatively unknown. It's sort of an interesting phenomenon. So what they went on to show in that paper is that KRAS functions as a dimer. So two KRAS molecules interact and then activate downstream signaling. And what they found was that that dimerization was important for that tumor suppressive effect. And when you took these cell lines which have low endogenous levels of wild-type KRAS, and you put wild-type KRAS back into them, which is that red line, the cells grew much more slowly. When you put in a mutant that could no longer dimerize with the activating mutant protein, those cells grew as if they didn't have any additional wild-type KRAS. In addition to that, what they found was when that scenario happened, when you inactivated or deleted the wild-type KRAS allele, those cells became more sensitive to targeted therapy, in particular MEK inhibition. So in the white bars are the parental cell lines, which are sensitive because, again, they have low endogenous levels of wild-type KRAS. When you put wild-type KRAS back in, they became resistant, and when you put in the dimerization mutant, they became sensitive once again, suggesting that even though these tumors may behave more aggressively, perhaps they're amenable to additional targeted therapy in the clinic. Okay, this is my favorite paper, mostly because I'm an author on it, but I think the science is really good too. So this is a paper where we were looking at immunologic mechanisms of anti-cancer therapy, particularly targeted therapy. And what we were really trying to do is target RAS mutant lung cancer using a combination approach. So what the thought was is KRAS, one of the things that KRAS does is it increases levels of cyclin D, which then bind to CDK4, which phosphorylate our RB and drive the cells through the cell cycle. So we thought if we could drive down cyclin D using a MEK inhibitor called trametinib, and then couple it with palbociclib, which is a CDK4/6 inhibitor, we could create synergy in that protein complex and inhibit the growth of cells. And we found that that was actually the case. You can see the cell viability experiment there in the bottom right. And that efficacy was dependent on functional retinoblastoma protein. And then when those cells were treated, they didn't just stop growing. They entered this state called senescence, which is we think is an irreversible cell cycle arrest, and then the cells secrete this host of cytokines. What we thought was interesting is when we treated cells with that drug combination, even though the drugs themselves are cytostatic, they don't kill tumor cells, they just prevent them from growing. We saw tumor regressions in mice with competent immune systems. So we did a whole bunch of experiments to figure out exactly why that was, and what we found is that it was related to immune surveillance, particularly with NK cells. So NK cells are part of the innate immune system, so they kill non-self cells without antigen-related activation. So when we treated the cells with both drugs, which are the waterfall plot shown in blue, this is a mouse model of lung cancer, the tumor shrank. When we depleted NK cells, which are the gray bars, with an antibody, we no longer saw drug efficacy. And similarly, when we depleted cytokines or other cell surface markers that we think are important for that response, we saw a similar thing where the efficacy of the drugs was minimal. So that led us to this model where we treat cells with cytostatic targeted therapy. The cells stop growing. They secrete inflammatory cytokines like TNF-alpha and IL-15, which recruit these immune cells, particularly NK cells. The NK cells come, they inspect the tumor cells, and they kill them. And this suggests a potential role for combining these type of agents with immunotherapy and other strategies. So in closing, I'll insert a plug for our the AATS International Thoracic Surgical Oncology Summit, which is going to be held this September in New York. We had a great program last year, and we're looking forward to another fantastic meeting this year, and I encourage all you to attend that. So.